Looking at this quarantine map of Southern California, agricultural epidemics like Huanglongbing spread rapidly across critical crop zones. Standard protocols require farmers to clip suspect leaves and mail them to centralized labs, but perishable plant tissue degrades quickly during transit, which routinely leads to unsuccessful identification and false negatives. To bypass the mail entirely, researchers developed this 3D-printed handheld microhomogenizer. It performs nucleic acid extraction directly in the field. This workflow diagram highlights the time savings. A benchtop lab protocol takes hours of manual grinding and refrigerated centrifugation, whereas the highlighted green steps show the handheld device producing ready-to-use DNA and RNA in just 12 minutes. Bringing the diagnostic timeline down to under an hour gives farmers the specific data they need to remove infected trees immediately, stopping outbreaks before they compromise neighboring groves. This cutaway diagram reveals the modular design. The lower section houses a reusable electric motor, coupling with interchangeable single-use processing chambers on top. Focusing on the top unit, the cap holds untreated cellulose paper discs, while the lysis chamber below houses the mechanical blade assembly. The field process starts by tearing a suspect citrus leaf down the middle. Plant pathogens concentrate in the vascular tissue, so you isolate and remove the midvein. You must finely mince that midvein with a razor. Large fibrous segments will jam the rotors. Load those small tissue fragments into the lysis chamber and add a chemical lysis buffer to help break down the cell walls. Power on the motor. The blade forces the tissue against the textured internal knurling of the chamber, mechanically pulverizing it into a crude lysate in exactly 1 minute. Tough plant cell walls usually require freezing with liquid nitrogen and heavy mortar pestles. Combining fine chopping with high-speed mechanical maceration forces the cells to rupture and release their nucleic acids right there in the grove. With cells broken open, flip the device upside down, forcing liquid lysate against the untreated paper discs inside the cap. During this 5-minute window, a specific biochemical capture takes place. The cap contains small, untreated cellulose paper discs. As the lysate rests against them, the paper acts as a sponge, extracting and binding the total nucleic acids from the liquid. This passive gravity soak effectively isolates the microscopic genetic evidence of the virus, separating it from the heavy, useless plant debris, and replace it with a clean wash chamber filled with a tris HCl buffer. Shake the device gently for 2 minutes. This clears away the remaining plant proteins and debris, leaving only the captured DNA and RNA trapped in the cellulose fibers. Remove the wash discs with sterilized tweezers and drop them into a clean tube containing a final elution buffer. While this field method yields slightly lower total nucleic acid concentrations than heavy laboratory centrifuges, testing confirms the captured amounts are consistently high enough to deliver 100% diagnostic accuracy. In 12 minutes, the raw, perishable leaf tissue has been successfully transformed into stable, isolated genetic material, ready for testing or transport. From here, you have 2 logistical options. If you want a lab to run the final analysis, simply let the paper discs air dry. The genetic material is now stable enough to be shipped via normal mail at room temperature. Or you can advance straight to testing right on the farm using the eluted liquid for a rapid RT lamp assay. These tubes contain a freeze-dried reaction mixture. Stabilized into these pink clusters, these reagents don't require cold chain transport to stay viable in the field. Pipette the eluted sample into the tube to rehydrate the mixture. Then, incubate the tube in a portable block heater set to 65 degrees Celsius for 35 minutes. Looking at this test tray, the final results are clearly visible. A sample that remains pink is negative. If the pathogen's RNA is present, the amplification process drastically shifts the pH, turning the liquid bright yellow or orange. This distinct colorimetric shift allows growers to achieve laboratory-grade certainty using only a portable heater and the naked eye. Adding it all up, 12 minutes for extraction and 35 minutes for incubation. This workflow graphic maps out the complete capability, moving from a suspect leaf to a definitive diagnosis right inside the greenhouse or grove. Because the motor base is reusable, you simply swap the top processing unit, load a new blade and paper discs, and immediately test the next tree. By compressing complex laboratory diagnostics into a portable 47-minute protocol, agricultural workers gained the immediate intelligence required to quarantine outbreaks and protect global crop yields.